Next start kori abar tane notun start beginning arma korbo erokom. Today I will discuss the disease of maize in the now start the northern leaf blight. Northern leaf blight is a pathogen is asexual is a Helminthosporium tarsicum অথবা exar o hilam tarsicum till sexual stage is Cystosphaeria tarsica. The most important symptoms is generally these starts small elliptical spots on the leaves, grayish green in color, water soaked lesion, turn greenish with increasing age, and increasing age the spots also bigger in size and spindle shaped. Individual spots usually 3 by 4 wide and 2 to 3 inch long. Spores of the fungus develop abundantly on both sides of the spot. Heavily infected field present a scorchy appearance. The disease is recognized by lobed elliptical spots, grayish green tan color. Fully expanded spots may 1.5 inch by 6 inches size. These lesions appear first on the lower leaves, and as season progresses, lesion may increase and all the leaves are covered with the spots. The plants look dead and grey. The spots produce the pathogen the conidia of the conidia 3 to 8 septate and spindle septate. And this pathogen survive in less warm and wet conditions. Continuous monoculture increase the pathogen can survive, and reduced tillage also favorable for survival of the pathogen. The disease cycle in case of this is the healthy leaves, the pathogen in the infected debris where the fungus and mycelium overwinters and husks on the other plant parts. The spores, these spores spread through wind, rain or leaves, attack produces symptoms to new leaves, and from the infection and symptoms development and from both sides of the leaves spores are produced. These spores are spread through leaves to new host and produces disease. This is the disease cycle. The disease management, use of resistant varieties, general sanitation, destruction of crop debris, rotation with non-host species, and spray Gineb or Manep 0.25% at 10 days interval. Also seed treatment with carboxin 2 gram per kg followed by the 3 sprays of Mancozeb 0.25% at 30, 40 and 50 days after sowing. Carbamazepine and Mancozeb mixture also used 0.25%. Metrium and pyrachlorostrobin by Cabri top 0.3% you can use to reduce the disease. Next is southern leaf blight. This is also caused by Bipolaris Maize, and this is also Helminthosporium Maize. Now it is called Bipolaris Maize. Sexual stage is Cogliobolus heterostrophus. The 3 physical races of the Bipolaris Maize: race T, race 0 and race C. Race 0 causes long tan lesions and that have brown borders on leaves. Race T and C are known to be specifically virulent to corn with cytoplasm of male sterile T and cytoplasmic male sterile C respectively. Lesions on leaves caused by race 0 of the fungus are elongated, tan color between veins to 1 inch long between the veins. Lesions produced by race T, which was very prevalent in early 70s, tan spindle shaped elliptical with yellow greenish chlorotic halo. Race T lesions often have dark reddish brown border and may occur on all parts of the plant. These are the spores of the Bipolaris origin, Bipolaris Maize. These are the spores of the Bipolaris Maize. This is the 12 septate. The septation of the spores is 12. Disease cycle, infected crop debris, fungal overwinters. These spores are produced in spring. Wind and rain move to spore, attack to the plant, produces symptoms. From these symptoms, both sides of the symptoms produces spores. These spores sprays by wind and others, rain splashes and attack to the plant and new plant and produces disease. This is the disease cycle. A predisposing pathogen survive warm temperatures 70 to 90 degree Fahrenheit and wet conditions, monoculture and tillage. Use of resistant variety, crop rotation in non-host species, sanitation, destruction of crop debris, and spray manage and Gineb 0.25% at 10 days interval can reduce the disease. Another disease, stalk rot of maize. It is also caused by two pathogens, Cephalosporium acrimonium and Fusarium stalk rot. Infection caused by Cephalosporium acrimonium becomes apparent when maize has reached the draft stage. One of the first symptoms, the purpling of the leaves and stalks. Characteristic symptoms, the restricted blackening of the vascular bundles in the stalk, shading of the intermedial pith region, and blackening of the vascular bundles extends through several internodes. Symptoms caused by Cephalosporium maize appear only after the flowering stage. Plants start wilting septically, giving a dull green appearance of the leaves, which later dry up. Lower internodes turn discolored, become reddish brown, sunken, soft, subsequently becomes dry and yellow, hollow. When this disease stalks are split open, reddish brown vascular bundles are seen. Barren plants, excessive tillering, multiple years are the other diagnostic symptoms. Cephalosporium acrimonium, Cephalosporium maize, these fungi are found wherever corn is grown. Both the pathogens survive in soil, plant debris and seed, overwinters in crop residue. Dry, warm weather, insect injury cause infection more, environmental stress and other diseases, high temperature, low soil moisture favors disease. Hybrids that are resistant to other stalk rot disease. Use hybrids, minimize all crop stresses, crop stress by application of fertilizers or with water stress or other stress conditions, control insect and plant population, use proper plant population. Another stalk rot caused by Fusarium stalk rot, usually distinguished from those other stalk rot diseases. Rot normally begins soon after pollination, becomes more severe as the plant matures. The leaves turn from a healthy green color to dull green. Wilted plants are remain standing, dry. Small dark brown lesions develop in the lowest internodes. You see, when split open, the stalks may reveal a whitish pink to salmon pink pith discoloration, confused with the red color of the gibberellic stalk rot. Premature senescence, lodging due to disintegration of the stem. Shading of the internal stalk pith may occur when squeezed between the thumb and index finger. The lower nodes, the stalks become collapses. These are the symptoms of the Fusarium stalk rot. Fungus survives in crop residue in the soil or in the surface. It may infect maize stalks directly through wounds or caused by the insect or nail. Spores may be sparsely dispersed into leaves and washed down into leaf in sheath and infect the nodes. The fungus commonly seed-borne by the source of inoculum believed to be less important, air and soil-borne inoculum. Fusarium verticilioides is most common in warm and dry areas. Fusarium root and stalk rot typically occur in complexes and other root stalks such as Gibberella, Diplodia and Coletorica. The Fusarium pathogens, Fusarium molliforme, Fusarium proliferatum and Fusarium subglutinans. These fungi are found wherever corn is grown, overintense crop residue, dry weather, warm weather, insect injury cause infection more, environmental stress and other diseases also increase the disease. The most important management is cultural control. Hybrid varieties which are resistant also can be grown. Plant remains less stress condition can reduce the disease. Less stress condition means use of high nitrogen, low potassium, high moisture is the mid or late season after dry, early season, moisture stress early in the season and during grain field, high leaf disease incidence. Physical damage that creates wound allowing the pathogen to enter such insect damage or hailstorms may also predispose maize plant to stalk rot. Where possible, harvest early to avoid yield loss due to